Assalamu alaikum dear students. I hope you are well by the grace of Allah with your friends and families. Welcome to our today's online class. First of all, I would like to introduce myself. This is Sifat Firdausi Shaun, lecturer, Department of Biology, Milestone College, Uttara, Dhaka. In your previous class, you learned about virus. Today, I will discuss about bacteria. So, dear students, let's start our today's online class. What we will be learn from our today's lessons. First of all, definition of bacteria, invention of bacteria, and characteristics of bacteria and the structure of bacteria. Lastly, we will know about the differences between virus and bacteria. Now, I will tell about the definition of bacteria. What is actually bacteria? This bacteria words is come from Greek word. Bacterium means is equal to little rod. Bacteria are unicellular, microscopic, prokaryotic organisms which are generally chlorophyll-less and covered by complex cell wall and mainly reproduced by binary fission. So, this is our definition of bacteria. We know about the bacteria. Bacteria is one kind of organisms that cannot be seen in the naked eye and requires microscope for their identification, are called microbes or microorganisms. Our chapter names also microorganisms. Bacteria are the first seen microorganisms and first life of the universe. That is an accepted opinion of the scientists of the world. Now I will tell about the invention of bacteria, and the most important thing I will tell about the invention of bacteria is actually so long, but I will describe in a short form. Antony von Leeuwenhoek in 1675 first observed small rod shaped organisms in the water drop collected from the rain water. While he examined it under the simple microscope designed by him, he called these organisms animalcules or small animals. For this reason, Leeuwenhoek is regarded as the father of bacteriology. After this, German scientist C.G. Ehrenberg in 1829 called these small organisms bacteria. Louis Pasteur, a French scientist, contributed a lot in establishing the new branch of biology as the bacteriology. Pasteur is regarded as the father of modern bacteriology for his contribution to the subject. So, this is our actually in a short form, this is our invention of bacteria. Now, I will describe the habitat of bacteria. Bacteria is present everywhere of this earth. Water, soil, air, even in the living and non-living bodies, in organic substances, in hot springs, extreme deserts or polar iceberg, etc. In nature, bacteria can grow in very cold like minus 170 degrees Celsius to high temperature like 80 degrees Celsius. Some bacteria live as parasites within the plant and animal bodies, and others live there as symbiotes. Normally, bacteria grows abundantly in environments rich in organic materials. For this reason, huge number of bacteria is found to be present in the cultivated soil and waterlands rich in organic materials. Bacteria is also found to be present in the air, but not in very high altitudes. So, this is our habitat of bacteria. Now, I will describe the characteristics of bacteria. All types of bacteria are microscopic, tiny and simplest organisms. So, first characteristics, number 1, bacteria are unicellular but more than one cell may form colony. Number 2, they are prokaryotic, that is cell wall organized nucleus is absent in the cell and a circular DNA molecule represents the nuclear material which remains present in the cytoplasm. Number 3, all the cell organelles are absent except the ribosome, that is 70S types. And then number 4, generally chlorophyll is absent, but if few are autophytic, that means they can produce their own food due to presence of a green pigment, bacteriophile. And then number 5, cell wall is double layered and main component of cell wall is peptidoglycan or mucopeptide. Sometimes a very complex type of cell wall is formed by aggregation of polysaccharides, morabic acid, teichoic acid, etc. And then number 6, generally reproduces by binary fission, that is amitosis. Amitosis means what? One mother cell divided into two daughter cells directly without any complication, that is called actually amitosis. Number 7, in some species flagella or pili may be present outside the cell wall. Genetic recombination may occur in a special process called conjugation. Number 9, most of them thrive energy by oxidizing inorganic salts. Number 10, they may be aerobic, anaerobic or obligate aerobes in nature. Now, I will draw a bacteria and I will show you the structure of bacteria and also I will describe the structure of bacteria. Of a typical bacterium, and this is the figure of a typical bacterium. Here as you can see, this is our cell wall. Now I will describe about cell wall. Below the capsule, this is our capsule, below the capsule cell wall is present. This is comparatively complex covering layer around the cell which is rigid, thick and elastic. This is the cell wall. Depending upon the species, cell may be 10 to 25 micrometer thick. Its principal component is mucopeptide. Polysaccharides, proteins and lipids may also be present in the cell wall. This is our cell membrane. So, this is our cell membrane. It is the thin living membrane below the cell wall and composed of lipoprotein, that is lipid and protein is called lipoprotein. Cholesterol is absent in the cell membrane of bacteria. As the membrane is semi-permeable, it regulates flow of soluble substances in and outside the cell. It contains various enzymes in photosynthetic bacteria. Cell membrane remains folded and forms thylakoid-like structure for holding the pigments. Till now I described the cell wall and cell membrane. Now I will go to the cytoplasm. This area is called our cytoplasm. In the cytoplasm, nucleus, basosome, vacuole, ribosome, all the things are present in the cytoplasm. So, what is cytoplasm? Cytoplasm is the granular homogeneous aqueous solution within the cell membrane. It is generally colorless. Within the cytoplasm, all the things like I said, vacuole, stored substance like carbohydrate, fat and protein and different minerals are present. So, it contains the substance like plasmid bacteria. Now, I will describe this. First of all, I will describe the ribosome. This is our ribosome. We know that this is the protein factory of the cell. Here, ribosome is small granular structure consisting of RNA and protein. Bacterial ribosome is 70S in size rather than vacuole. Here, this is our vacuole. Vacuoles are small sac-like structures containing cell shape within the cytoplasm. It maintains hydrostatic balance of the cell. Now, I will go for the volutin. So, this is our volutin. Bacterial cell stores food material in the form of volutin within the cytoplasm of younger cells and within the vacuole of older ones. Now, I will go for the mesosome. This is our mesosome. The cell membrane folds inwards in some places and forms sac-like mesosomes. Several respiratory enzymes remain present. Now, I will go to the nucleate or pseudonucleus, where we can see this is our nucleate or DNA, or we can say that pseudonucleus, that means false nucleus. Bacteria are prokaryotic and the nuclear substance contains only chromatin or DNA, we know that. It is a double helical circular DNA molecule. Here, as you can see, this is the circular DNA molecule, much larger than the diameter of the cell and remains repeatedly folded at the center of the cell. So, this is our nuclear material. Now, I will go for the plasmid. This is the very important thing. This is our plasmid. In the cytoplasm of some bacterial cells, one or more small self-replicating circular DNA molecules may be present in the addition to the chromosomal DNA. These extra chromosomal circular DNA are known as plasmids. Plasmids are self-replicating and contain a few genes, that is E. coli have plasmid. Now, I will go for this flagellum. Here you can see this is our flagellum. Some bacteria contain long whip-like movable outgrowths outside the cell wall. This type of moving organs are known as flagella. I will go for pili. Here you can see this is our pili. In the plural form it is called pili, that's why I show you two pili in the figure, and pili or its another name is actually fimbriae also. In some gram-negative bacteria there are present numerous small rigid hairy structures projecting the cell wall. These outgrowths are called pili. The bacterial cell remains attached to the host cell by the pili. They are made up of a special protein known as pilin. Conjugation tubes are formed from the pili also, which are generally known as sex pili. So, this is our structure of a typical bacterium. I hope you understand the structure of a typical bacterium and you will draw in this way. If you draw in this way, then you will get full marks in your exam. In your previous class, you learned about virus and today you learned about bacteria. So, you have to learn the differences between virus and bacteria. Now, I will describe the differences between virus and bacteria. Differences are size, bacteria larger, virus is smaller, and then cell wall. Bacterial cell wall is made up of peptidoglycan, viruses no cell wall is present, protein coat present instead. Ribosomes. Ribosomes are present in bacteria and viruses. In virus, ribosomes are absent. Number of cells. one cell, or and it can call as unicellular bacteria. Viruses have no cells and living or non-living. Bacteria, living organisms, viruses between living and non-living things. DNA and RNA. DNA and RNA floating freely in the cytoplasm in bacteria. In viruses, DNA or RNA enclosed inside a coat of protein, and the infection localized and the virus's infection are systemic. There are so many kinds of differences are present. You will learn and you will find out what is the actual differences. Now it's time for homework. Here, as you can see, this is a stem, figure A and figure B is present. So first question is A, what is bacteria, and number B, that is comprehensive based types of question. Write down the characteristics of bacteria, and number C, describe the structure of the organism B, and number D, analyze the dissimilarities of the A and B according to the stem. So this is our homework. Please submit your homework in your respective subject wise homework group. So dear students, I hope you all enjoyed our today's online class. If you have any query, please ask your subject teacher. Thank you for being with me. Stay home and stay safe. Take care. Allah Hafiz.